Okay, hi, good evening, everyone. So, any doubt in the last session, guys? Shri Charan sir, Sonali ma'am. Any doubt, Amit sir?
No, ma'am.
Okay. So, can anyone tell me what we did till now, the in the pipeline? Shri Charan sir. Amit sir, can you tell me? Okay, so this box plot also you can export in your folder. You can save this image as the PDF or the image. It's up to you. And it will be downloaded in that folder. And you can just give the name of that plot, saved. So yeah, we what we did, like after loading your libraries, we use the readFE function so that it can read all the FE metrics data that we have loaded, all the cell files. Then after reading the FE files, what we did, we want to see how the intensity look like, right? So for that, we were like use the box plot, and we have given the different colors for the control and the blue, like for the treated. Then what we saw, like in this figure, the all the median lines, it is not evenly distributed in this data. So that means there is some variability in our data, right? So here in the microarray, we are going to compare one sample with respect to another, right? So we can only compare when all the median line is equal for the control and the treated. And also by looking at this data, we can see there are some error or the biasness in our data. So to remove that, we have used one function, that is the RMA, which stands for robust multi-chip array, which it has what it did, like it has background correcting, normalize, it has just changed your intensity values into the log2, that is the normalized count of your data now, and also given the expression count. So after viewing this norm data, you cannot see what are the like expression values for your data. So for that, we are going to use the express function. Then after running this expression count, you can view and you will get the numbers. Okay, that is would be the normalized count, the log2 values. Then again, you can generate the normalized box plot. After doing that, you can see it, the median line is equal for the control and the treated. Then again, after that, what we saw, we calculate the t-test so that we can just generate the p-value, right? So in this R Studio, t-test is the user defined. Okay, it is not there in this R Studio. So we have generated the command, or you can see the code for the t-test. Here 1 and 2 is what? The control, and 3 and 6 it is labeled in the treated. Then we just printed the p value and we got the p value for this. Then after that, in order to see the p value along with your table, we just use the cbind option, and then we saw the p value in the table. Just wait. Okay, so what would be the next step now after the p value?
Log fold change.
Log to fold change. Okay, so that means the analysis plan you are talking about, right? Because
Yes,
already
ma'am.
these values are, ma'am, log 2 only values. Okay, these log 2 values. So along with that, the p value you are getting.
Yeah,
Now
okay.
we are going to calculate what? The differential gene expression, right? So before calculating the differential gene expression, we should know what is my analysis plan, right? So here I am going to choose what the treated versus treated versus control plan I am going to use. Because most of the time we want to see what is the change in the treated with respect to the control. So that is why I've taken treated versus control. So you already know, like in the cell file also, like we have taken two treated, right? And for each and every treated, we are going to find out what is the change with respect to the control. So now further, we are going to take what? The mean of the control. Okay. So for that, what we are going to do, we'll calculate the mean control. So how to do that? By combining all control together, and we'll take the mean of their expression. So we'll make, so I am taking one variable, that is the mean control, equal to, I'm going to use one function, that is row mean, and then you have to give your input file name. So this is now input file name, that is the combined p value, all the data it is present here. Then in this bracket, just load your data. Just let me write the command and I will explain you. Control 1. So I'm taking the row mean function. So this is one of the function which is used to calculate the average of each row in the matrix. So this is my input file name, combine p value. So if you have like run the R programming before in the R studio, so whenever we are calling the columns, that time you have to put before that comma, then give the column name. So you can see in the combine p value, I have taken this control 1 dot cell, then control 2 cell. So this I have loaded uh, in this, yeah. Then after that, NARM. So NA dot RM, what is that? A logical value which is indicating whether the missing value NA should be removed before the calculation. So why I have written true? True. So for example, like some of the values are missing in your data. So that missing values I have to remove. So that particular, like I'm specifying with the help of true. So it will remove all the missing value from your data. Got it, this command? And also you have to write C because it is in the combination. We are taking two sample. So that is why you have to give the C here. Mm. Yeah, so I've missed one bracket here. Done, all of you. Can we move ahead?
Yes, ma'am.
Okay. So here we have taken the mean value of the control, right? So I want like the mean values and all these values should be together in one table. Okay. So now again, I am going to use the cbind function for combining all these two tables. So I'm using one variable again, that is all underscore table equal to uses cbind function. So I'm going to combine these two files. First, this is the combine, then give comma, And this is in this data mean control, you are having the what? The mean of control. So now just paste here, then run it. So now you can view this. All table. You can see all the control values, p value along with the mean of control. Clear? Any doubt, guys? So now can you tell me what is the fold change formula? Log 2 treated minus log 2 control. So this is the formula of the fold change. So we have calculated the p value, which is going to tell you the significance of your genes. Then next we have taken the control means so that we can compare the treated all the treated sample with respect to your mean control, right? So now further we are going to calculate the fold change. And for the fold change, so for example, if you have taken the treated versus control, that time the formula is log 2 treated minus log 2 control. If your plan is control versus treated, so it will be like opposite. So the formula will be log 2 control minus log 2 treated. Then now we'll make the formula. Okay, so take one variable. So for example, I'm taking this fold underscore change, like equal to change. Now then I will load the table. All table. Why? Because all the input it is there in this data now, right? So just write here all table. Then in the all table, I'm going to call first the log 2 treated values, right? So you can see if you open this all table, so treated values are listed here in this third column, 4, third, 4, 5, 6, right? So you have to write here, comma, 3 underscore 6. Just give me a second. So fold change all table is my file. In this all table, I'm going to call first the log to value of the treated, then minus, put the brackets, and now I'm going to create what the values for the control, mean of control. Okay. So all table, I can just check it. 1, 2, 3, 4, 8. In the eighth column, you are having the mean of control. So just type here 8. Okay. Is it clear this command?
Yes, ma'am.
And also this, remember that in this table, these are the log2 values for the control 1, log2 values over the control 2. Now you can view this, and now you can see this is the fold change values. Okay. But you can see the names of the fold change is like it is wrong here. Can you see here? Because I've taken what the log t1 minus control, then log 2 treated 2 minus log c, right? So we are going to change the name of the headers. So for that, what you have to do, just write here call names, then load your file, then equal to combination. So I'm going to label like this, treated 1 versus control. So now I'm going to change the header with these names. Okay. Can you just try in your system? Now you can see the labels are changed. You can see it is the fold change of your treated one versus control. Likewise, this is we are going to do the analysis for this. Clear? Any doubt? Is it done, guys?
Respect.
Now again, I want this fold change values together in this all the table file, right? In the all table, your all the data it is present, but not the fold change here. So again, I am going to use what the cbind option. So I will just write here fold. So I'm just taking one variable. You can use any name and just call the cbind function. And so I'm going to combine this all underscore table, comma, this fold change. Then run. Then now you can view, you can check whether it is present or not. Now you can see along with the log2 values, p-value, the fold chains are also present in one file. So all the data it is present here in this fold change table. Any doubt till here? Actually, this ours today is very sensitive. So you can see like I missed one comma. So it was throwing one error. So be careful with the codes also. Okay. So in this step now we are going to export this fold change table where all the values are present. So how to do that? You have to use one function, that is the write dot table. Then put your input file name, that is the fold change table. So in which file you want to make, you can just write a file equal to, we want in this format, in the Excel format. You can give your name of your file. So I've labeled that microarray treated versus control. The next thing I have given code is equal to F. So what is that? Like again, we are going to instruct the R programming not to quote the string. So this is very useful whenever we want to create a plain text file without unnecessary quotes, such as maybe CSV file. Then next is the step, which I have denoted with the slash and the T, which is indicating the tab character will be used as a separation here. Then call names equal to NA. And again, we are telling the R programming not to write the column names. So this is useful whenever you want to append your data to an existing file without adding any new header row. Okay. So now you can run this and you will get the microarray file. Yeah. Is it clear? Any doubt in the pipeline? Anything you want to know from this pipeline? Are you able to execute till here? Shircharan sir, um, Sonali ma'am, Amit sir.
Mean what a fold change value indicates.
See, why we have calculated the fold change? We have discussed that day also, but again I will tell you. So whenever we are comparing one sample with respect to another, that is what the fold change. Okay. So this log 2 fold change we are calculating. Why? Because whenever we are taking only the log 2 values, they will generate what? The values in the form of positive and the negative values. So positive we are going to indicate with the upregulated values, and the negative will indicate the downregulated genes. Okay. So whenever you are going to calculate the differential gene expression, you have to calculate the fold change. Then only we can compare one sample with respect to another. Okay.
Yes, ma'am.
Because simply calculating the log 2 values, now you are not able to tell me whether it is the upregulated and the downregulated. So after calculating the fold change, it will be very carefully, like you can notice in your data also, these are my negative values, these are my positive, so you can easily interpret your data. Okay. So what you can do, this first column is my prob id. These are my log 2 values of the control 1 and the control 2. And these are also, again, these are log 2 values. This is the p value, which will tell you how significantly the gene is expressing. And this is my mean of the control. Okay. and this is my fold change values. Now what you have to do, just copy the entire data and paste in next sheet. So here I'm going to rename the file T1 versus control. Just delete this. Likewise, you can generate for other also plan, because total 4 plans will be there. Treated 2 versus control. Again. So you can separate all the plan in this 4 file, and then further analysis we will see tomorrow. And then you can save this. Any doubt? So everyone can separate this fold change value for each and every plan. And next part we'll see tomorrow. Amit sir, Shricharan sir, Arya, Yash, what about you guys?
Done, ma'am.
Yes, ma'am.
So is it clear till here? Till the export of the data?
Yes.
Yes, ma'am.
Okay. Okay, then we'll see you tomorrow and we'll see further analysis.
Okay, ma'am.
Okay then. Yeah. Bye bye.
Okay, thank you.
Thank you, ma'am.
Okay. Bye bye.